Hi everyone, this is the last part of chapter 16. This is 16.5 and this is on riboswitches. And just a little hint if you need some help on this, I believe this is what Rachel, our embedded tutor, did her mini lecture on last semester or last year. So if this is confusing, she'd be a great person to ask as well as I am. So let's talk about what riboswitches are. So riboswitches were discovered actually quite recently within the last 20 years, and they're a type of regulation that we find in both prokaryotes and eukaryotes. So in this type of regulation, the RNA, the actual mRNA, can exist in two different conformations. One of the conformation is what we would call active, meaning that it's going to go through transcription or translation, and one conformation would be considered inactive, which then either transcription or translation would not occur. And so the switch from one of these conformations to the other actually relies on a molecule that binds with the RNA, and generally it's a either product or some sort of substrate that is involved in the actual mRNA product. So we'll talk about that with a couple of examples. So riboswitches, the two that we're going to go over at least, regulate transcription and translation, although they can also play a role in regulating RNA stability. So they can actually make an RNA less stable if the organism is not wanting it to be translated, which means that it gets degraded. And they can also do splicing. So when we talk about eukaryotic, when we look at eukaryotic transcripts, we know that sometimes they do splicing, and riboswitches can do that as well. So this is giving a quick summary of these different ways that this can happen. So with the regulation, the types of regulation, we can have transcription. And in this first one, it means that the 5' region of the mRNA can exist in a conformation that either forms a row-independent terminator, which would cause attenuation of that transcript, and the other conformation then does not form an early terminator and is completely transcribed. So it can actually shut down transcription. These riboswitches can in this part. For translation, the 5' region of that mRNA may exist in a conformation where the Shine-Dalgarno sequence can't be recognized by the ribosome. Remember, the Shine-Dalgarno region sequence will actually bind to its complementary rRNA in a ribosome. But if it is made inaccessible, then it won't actually be able to go through translation. And then in the other conformation, it would make that Shine-Dalgarno sequence accessible, which allows it to bind to the rRNA in the ribosome, and then the mRNA could be translated. As I said, it can also play a role with RNA stability. One of the mRNA conformations would be stable, whereas the other conformation would act as a ribozyme, meaning that it causes itself to self-degrade, so start to break itself down. And then in eukaryotes, we can have a pre-mRNA conformation that can be spliced one way, and in another conformation it would be spliced in a different way, which would mean that either it's not going to go then through translation, or maybe it changes the protein that's being made. So let's look at a riboswitch that can regulate transcription. These are fairly well studied in bacteria at this point. So the one we're going to look at is called thiamine pyrophosphate in Bacillus subtilis, which is a bacterial cell. And this is a really important vitamin, because it plays a role as an essential coenzyme for the citric acid cycle. So when levels of this particular vitamin are low, so when you have low TPP, there's a stem loop that forms called an anti-terminator, and this will allow the cell then to make more TPP, so it'll allow transcription to occur. However, if a lot of TPP is present in the cell, it will actually bind to the mRNA early on, which causes a change in the secondary structure. It will actually put in a terminator loop early, which stops transcription and inhibits the production of enzymes that would make more of this TPP. And I think I have a picture of this. So when we have low TPP, so we need more of this particular vitamin made, what ends up happening is that we have the 5' end, we have the mRNA, and we have this what is called an anti-terminator stem loop, meaning that transcription can occur to the end of this particular operon. However, it can switch when we have high levels of TPP. There's a section of the rRNA that TPP will bind to. It changes the conformation of this mRNA, which now what was our anti-terminator stem loop has now become a terminator stem loop. And what will happen is that we'll get stoppage of transcription at this U-rich sequence right after the terminator stem loop. And so that means that we will not have a full transcript, and we would not imagine that it could go on to make that TPP when there's high levels of it already. So again, that's kind of that negative feedback we talked about earlier in other types of regulation. The second place that we can see a riboswitch regulating is in translation. So this is actually studied in E. coli, and in this case, this is thiamine pyrophosphate, and this particular operon codes for 2 enzymes for making TPP as well. Okay, when the TPP levels are low, the 5' end of the mRNA will fold into an a stem loop called the Shine-Dalgarno anti-sequester. What this does is it makes that Shine-Dalgarno accessible. So remember, the Shine-Dalgarno sequence has to be accessible to actually bind to ribosomal RNA in the small subunit of the ribosome. So we would need to have this exposed or accessible so that translation could occur for this. But we could get the opposite, that when TPP levels are high, they will bind the RNA, which make it so that this Shine-Dalgarno is sequestered or blocked from binding, which then would block translation. So this is a picture of what this looks like. So low TPP, we have our 5' of our mRNA, we come around and we have the Shine-Dalgarno sequence followed by the start codon that are exposed. These then can be accessed and bound to the small subunit of the ribosome, and translation can occur. However, if we have high levels of TPP, it changes the conformation, and what ends up happening is we get a stem loop with a Shine-Dalgarno and start sequence. This can no longer, or is no longer available to bind to the small subunit of the ribosome, and therefore translation would not occur. It would be inhibited. So just a quick checking in for a riboswitch that controls transcription. What would the binding of a small molecule such as TPP control do? So you want to think about, with riboswitches, what is it that these small molecules can do, and think about the conformation of the messenger RNA. So that's the end of chapter 16. If you have any questions, please do let me know. I'm happy to make some videos of maybe some of the more complex concepts in here, or put up videos that are on the internet that might be helpful. Thanks.